Hey guys, this is Mr. Kelbley, and welcome to our video on translation, which is the process where RNA is finally converted into proteins, which in turn build us. This is going to be the first of our videos that you're going to be watching at home, so I have included a slide here with a few reminders for you. If you need them, please feel free to use them. I'm not going to spend the time talking through them right now. So, to review what we know so far about DNA and RNA and how that all works, think to yourself, copying DNA is known as what? I want you to come up with an answer. Okay, copying DNA is known as DNA replication, right? So that's when you take one DNA double helix and you copy it into two new DNA double helices. Next one, converting from DNA to RNA. So taking the information from DNA, putting it in RNA, this is known as transcription. Finally, how do we finish the process and get from RNA to proteins? The answer is translation through gene expression. So what is gene expression? This is basically just the term that discusses the whole process, the entire process of how information is taken from DNA all the way used to make proteins. So starting with the DNA, going through transcription, then translation to get to proteins. On your guided note sheet, you will see that you have a diagram. The process of going from DNA to RNA, as we already know, is called transcription. The process of going from RNA to proteins, we should already know, is called translation. That's what we're going to be going into a little more today. So translation is the process that uses the information from RNA to build amino acids together to make proteins. So if you remember all the way back from, I believe it was October, when we learned about amino acids, which are the building blocks of proteins, we learned about those organic compounds. It's all starting to come back now. Translation takes place at the ribosomes of the cell, not in the nucleus. This is extremely important. Proteins are built at the ribosomes, and this is where translation happens. Translation has a few main steps. First, RNA goes to the ribosomes. So the mRNA leaves the nucleus and travels to the ribosomes because that's where proteins are made. Second, there's something called tRNA. This is T for transfer RNA. And this matches up with the mRNA codons. Remember that a codon in mRNA is 3 base pair letters together. So, for example, AAG could be a codon. Each tRNA with 3 base pairs attached has an amino acid attached to it. This is step number 3. These amino acids attached to each tRNA end up connecting in a long chain called a polypeptide, to form a protein. So here's a picture of what translation looks like. And I want to take a few things to look at here. So as you can see, there is a group of 3 bases, 3 nitrogenous bases here on this mRNA strand. This group is called a codon, right? That's what that means. Attached to this is the tRNA. And the tRNA, the matching base pairs, those are known as an anticodon. The tRNA has an amino acid attached to it, as you can see up here. And as each tRNA comes in and attaches to the mRNA, it puts together these amino acids in a long chain like this. And this polypeptide chain, that is a protein. That's what that means. Last but not least, notice that this all takes place here at the ribosome. So that's all well and good. But how do we use the information that we have in DNA to determine what proteins we're going to have? That's what's really important, right? Because once we know what proteins we're going to have, that's when we can start to figure out what kinds of traits will be expressed, like hair color, like eye color, things like that. Again, each mRNA codon represents one amino acid. So every 3 base pairs represent 1 amino acid. So you can think of it this way. Codons are like 3-letter words. Remember, we've talked about the letters A, T, C, and G, and in RNA, A, U, C, and G. You put these letters together in different combinations to make words, and this is the language of genetics. These codons will be used to read something called a protein sequence table, which we're going to look at in just a moment. So this is a protein sequence table, and it looks extremely confusing. I can already tell you. But honestly, it's very easy to use. What you're going to do is look at the 3 letters of RNA, and you're going to use that to read the table. The first letter of the codon in mRNA, you're going to look for here. So let's say we have CAG. We're going to look for C first here. So this is C. That means it's going to be in this row. Second, we have A, right, for CAG. Where is A at for the second base? It's up here. A for second base. So we're going to look at CA and then G over here. CAG comes out to be glutamine. Glutamine is that amino acid, that nitrogenous base that builds this protein. So let's do one more together. We're going to translate the codon UAGCCU into amino acids. Those are actually 2 codons. So the first codon is UAG. The first base is U, the second base is A, and the third base is G. So just so you are aware, at the end of your packet for this week, that very last page, you have a full-page protein sequence table that you can use to make this easier for you. You can rip it out if you'd like. So base 1 is U. That means we're going to come up here. First base, you'll see U right up here. So it means it's in this row. The second base is A. So second base A, that means it's in here somewhere. And then finally, the third base is G. So we're going to look at third base, UCAG. UAG turns out to be something called a stop codon. And that's actually a signal telling the ribosomes to stop translating proteins, to stop translating mRNA. So that's not actually an amino acid, but that's another important one that we're going to need to be aware of. Again, there was a mistake here. If it were CAU, it would turn out to be histidine. My apologies for that. UAG would be the stop codon. So looking through how we got histidine for CAU, first base C, second base A, third base U, and that comes out to be histidine. What I want you to do and bring into class is figure out the second codon. So as you see here, down here at the bottom where my mouse says CCU, I want you to figure out what amino acid that would be. Alright, so thank you for watching this video. If you keep going for just a second, there will be a video of translation to show you just exactly what this looks like.
Translation is defined as the process by which messenger RNA is decoded to produce a chain of amino acids that later become a protein. A small ribosomal subunit positions itself on the mRNA molecule. Transfer RNA molecules carry specific amino acids that correspond to the tRNA anticodon. This anticodon is the code that allows the tRNA molecule to bind to the complementary mRNA codon. Next, the large ribosomal subunit associates with the small subunit and tRNA molecule to begin the translation process. As the ribosomal complex moves downstream on the mRNA molecule, tRNA shuttles each individual amino acid as specified by the code contained on the mRNA. Once the ribosomal complex reaches the final codon contained on the mRNA molecule, both the ribosomal complex and the newly formed amino acid chain are released to later become part of a protein.